other reason aside from what I wrote for like because I picked that one, there were less complications that could occur because I think that's the optimal one.
Right.
So that's why I only listened to like I said, maybe I could have come up with a third one, but there's also that kind of thing too.
Right.
Yeah, the only two I could think of was one that it's not like phosphorylated properly, and then two that it's not secreted properly.
I said the protein folding, and then I did both.
Oh, true.
So
It
maybe
could be protein. It could be protein folding.
Yeah, so it says
isolation. Probably happened correctly because it's the one.
Yeah.
But you're right that technically.
And I said, yeah, I said that it could not be like phosphorylated enough, and then the solution could be to like add genes to like phosphorylate. Like, I feel like all these design questions are like open.
exactly.
Could do different things. Yeah.
Yeah. I mean, ideally, it's just like, it's like a opportunity for us to get an insight of like how you logically think through problems. Like, yeah. But the thing is, at the end of the day, the way that we grade these things is like, how close are these students' answers to the answers that the teacher provides us? So there's like, and yeah, when it comes to these more subjective, open-ended design problems, it's difficult, and yeah, we need to have a more standardized way to
Wait, I actually do have a question on two, is when it says, the delivery systems for RNA therapeutics, the only two I could think of were viral vectors and then lipid nanoparticles.
For number two.
Yeah, for the delivery system. Because for
Right.
types,
So-
yeah, for
Yeah.
types I have four. Because I know that it's like there's a lot of types of RNA therapeutics,
Yeah.
but for delivery systems, those are the only two I have.
Yeah, I think two is fine. There's one other one. Wait, did you look at the slides?
Yeah,
You did? And there was only two on there?
from what I could find, yeah.
Okay. Do you have the slides up? The Kunzing slide? I'll look at it too real quick.
Oh wait, no, this. I think this is from the guest lecture.
The Zing, it should be in the English, yeah.
Yeah, well, there's a big slide that says delivery is therapeutic nucleic acid agents, and it's like viral vectors and lipid nanoparticles.
Yeah.
Like that's
Okay.
and then that's like what you got from ChatGPT.
Okay. Yeah, I mean that should be fine then.
Oh, is the other one polymeric nanoparticles?
One. That's the one.
Well, that's what ChatGPT gave, but I didn't know if like computers actually.
Right.
But yeah, I guess I can.
She literally copied this.
Yeah.
So yeah.
Yeah, that's what I do with the clinical applications. I just did all of the ones that like cancer therapy.
I really think that this homework is a lot of literally take it from the slide.
Yeah, that's why I feel like. Well, I feel like hopefully the final isn't too bad, but I feel like everything's like straightforward.
Yeah,
from this.
Yeah, I think the Zing's lecture was set up very clear. It was just really easy to understand. And I feel like it was pretty thorough with how, like, from beginning to end, he was like, okay, here's this thing I'm telling you about. Here's an example in a clinical application, and here's what we do. Like,
Yeah.
pretty thorough.
Okay. Yeah. Cool. Do you guys have any other questions, specific questions about the homework? What did you guys get for number 4? Oh,
Yeah.
was it?
So you.
Okay. All right. Okay. Yeah.
That was like. Murder. I guess. Yeah, like 9 and 10. But even those. Well, 9 and 10 was I kind of had the same answer for both, but like a little bit different. Because they were both that basically like using Raman. But I guess I did differently.
Do you want to discuss what you wrote? Or you think it's fine?
I think, do you wanna?
Yeah, well, so for 10, I basically just said using heavy water and like, using heavy water so that like basically you can label it and then you can see the amount of molecules like made with D instead of H,
Mm-hmm.
and then compare that. Basically use like well, okay, I think the confusing part about 10 was where it said from in a particular liver cell from a piece of liver tissue.
Mhm.
So I said that you could use like a sorting thing to make it like single cell and like a 96 well plate to get like that one cell, and then put heavy water in like one well.
Whoa.
And then put that into like a piece of liver tissue, because I didn't know if it was like just because it says in a particular liver cell, so I didn't know if that meant like one cell or if
Yeah.
that meant like a portion of the tissue is
Right.
changed.
Yeah, I mean, I think that's totally valid. That actually offers like an orthogonal experiment to I mean I think when I think when I thought about it, it was just like when we use Raman spectroscopy, when we take a biopsy from a mouse or whatever and cut out a part of its liver and we fix that to a slide and we use Raman spectroscopy, we can actually I mean I'm not 100% sure about this, but we can actually define each of the individual cells in the liver. We have that resolution just from the microscopy alone. So I think that yeah, I mean it's that's how I answered the question, but I think yours is totally valid too. And if we actually separate the cells based on its like sub liver cell types and we analyze how the metabolism is different in each cell type, no, that I think that gives us more information about that. So I think that's valid.
So I'm guessing like different experimental designs are okay as long as it's like the basis of ramen. Yeah, if you're using the
Kind
basis
of like
ramen.
yeah, use the heavy water and then use the ramen.
Okay.
Definitely, yeah. I mean, I mean there are other ways. Definitely there are other
Yeah.
ways, but it's like that starts to fall outside of the context of our class.
Yeah,
And yeah, if you, I mean, but then yeah, then you wanna then you're testing to see how lenient your TA is. Because it's like again, we have to come back to the rubric and be like, well, I don't see any mentioning of ramen, so
yeah.
and yeah, and depending on their mood, it's gonna either they're either not gonna take the time to really understand what you're saying or they are, yeah.
I basically, I didn't use heavy water, I just used like GFP to sort it. But I also separated the liver cells from the tissue I just said by enzymatic methods. I just left it at that.
Mm,
But then I used
to separate the cells. Oh, okay, yeah, that's fine.
And
Anyway,
then I used chemistry.
okay, yeah, I think. Okay, so okay, wait. So you do use heavy water, or can you? Okay, so okay. Heavy water I think it's water that has I don't know if it's a proton or a neutron or something like that, right?
Yeah, it's a neutron.
It's a neutron, yeah.
It has 2 neutrons instead of 1.
Yeah, something like that. Yeah.
It's like, because normally when you do like spectroscopy or spectrometry, like CH bonds like have a certain peak, but like CD bonds where like D is there instead of H has a different wavelength. So, or like has a different peak, so you can like track it, and it also doesn't interfere with metabolism. Because like if you do like alkyne tags, then that like it can't get metabolized. But if you just do like D tags, then it can. Because it basically the body like thinks it's hydrogen.
I think that's better than the GFP because the GFP is like so big.
Yeah, exactly. This is the crux of Raman microscopy. It's just like it goes back to the analogy that she had mentioned about what happens if you want to measure the dynamics of a ping pong ball, but if you're gluing on a basketball to it, then
Yeah,
it's going to the dynamics of it is it's
Yeah.
not going to represent its natural dynamics. It's going to be completely influenced by the basketball, and so it's like, yeah, I think the there is this, I don't know, necessity or desire in biology to create a technology that it is label free and in vivo imaging. Something where we can observe something in its natural state, but and identify these molecules as well without having to because the thing is one of the I guess contradictions in science is it's Schrodinger's box, right? It's like my how do I observe something without having my observations influence my outcome? And everything that we do in biology, because we put on that green fluorescent protein, it's in some way it's influencing our outcomes always. And so we're trying to get to this point where can we actually bring down the tag to in the case of deuterium, just a just a single proton. And yeah, can we bring it back down to that?
Yeah.
So that's the holy grail.
Also, like all the slides on that in her lecture, like all the figures are used from her paper. So I just looked up her paper and then she like explains it really good. And then in my homework, I put like screenshots from the paper of like kind of stuff that she did. Basically the same pictures that are in the slides. But yeah, that was helpful too.
So the thing about your answer with the alkynes, the chemistry, I don't think that actually addresses the question because I'm not 100% sure exactly how the click chemistry is involved here, but as far as I understand it, it's just like going to non-specifically add alkynes to, remove hydrogens on carbons with a triple bond, right?
I was thinking that the chemistry would react with the label, but not react with anything else.
Oh,
That's how I was thinking of it. But I think I was wrong when including GFP as well, because like I think that's like doubling up the labels for properties. But that was my understanding of it, but I don't know.
I don't know. I need to look into the mechanism. I don't know. So wait, so the GFP would be used to,
Would just be for the labeling, but it might not
so labeling what?
like being onto the I can erase it now, but
It was GFP, one of the labels in the slides that she said we could use.
maybe not in her the other slide, but I think I will look at that paper because she has those slides that are like it's like a series of slides that just talk about like the bioorthogonal or something related to it.
Yeah.
Yeah, I mean, I guess the thing that seems a little ambiguous to me is how are you using either the click chemistry or the GFP tags to specifically associate with the newly synthesized molecules?
Yeah, I think that's all I have, that the click chemistry reacts to the label and not reacting to anything else. And then we would use like Raman to visualize the.
Okay, but then how do you get the first tags, the GFPs, I guess, onto just the newly synthesized molecules? I don't know either.
That's why I think the deuterium is a better mechanism of doing that, because you can just add the deuterium water like at a certain point, and then you know like everything with D labels are new from when you added it. Right. And I also don't know if GFP like can specifically bond to like macromolecules or whatever, because like D just acts as hydrogen, and there's like hydrogen on like every organic thing, or not every, but like most. So,
Yeah.
but then it's still like the ramen, and then you can see the new stuff. So it's still like the same imaging process.
Yeah. I mean, it's if you wanted to use GFP, you would have to have the GFP be a part of a fusion protein.
We're
And
just adding more and more stuff.
yeah, and then you would have to put that into the genome. And then you would have to activate the, well, I mean, yeah, you can do that. And then you maybe it would be under some kind of like inducible promoter. And then you could turn on the expression of that one fusion protein for a given period of time. I mean that would work. That would work.
But I do think it takes away from ramen, because like you said, like we're trying to get to label-free mechanisms. So like GFP it's a method that works, but it's not the most efficient. So like you said, with like the basketball model, like we can use it, but it's not ideal.
Right. Yeah.
I'll look at it.
I mean, you can do this, but it's like there are a lot of metabolites, so you'd have to create like, I don't know, a lot of different you'd have to insert a lot of different fusion proteins into the genome of the, yeah, in order to get like all the different metabolites. Yeah.
That's all I have for now.
Okay.
Yeah, it is.
All right. Do you guys have any other questions? Do you guys want to go over the slides or something, maybe briefly? Or
Yeah.
Just like skim the it's fine.
yeah, let's do that. See.
Cause like you haven't, cause I remember in the midterm when we were going over it, like you took it already, but you haven't taken the final.
I haven't taken it yet.
Okay.
Actually, did she even send it to us? Actually, I think she did send it to us. I can maybe. Yeah, I haven't taken it yet. I'll probably take it over the weekend, and then maybe I'll do another discussion section on the weekend or something over Zoom. I don't know. I'll make an announcement.
Are you the TAs gonna do like practice questions like the midterm, like where you like do a set of like 3
Oh.
questions?
We haven't talked about that, but I can bring it up.
Professor, about a week ago or so.
Yeah.
And asked her like about if she was gonna do the final slides, which she's done, and if she was gonna post questions, and she did say she would. but-
Okay, this is what I'll do. I'll take the midterm, and if I feel like it is like not easy, I will definitely make questions. But if I feel like it's like most of it is in the slides, and I will
Oh, okay, okay,
I'll
yeah,
do
let's
it.
do it.
Yeah, I'll do it like that.
Would you be willing to give an outline of questions?
Outline of
Questions, but like,
yeah,
whatever you're willing.
yeah, I will. Yeah, I will. Yeah, maybe I'll do something general like last time, or I just like ask like super general questions. Maybe I'll do something like that. I don't know. We'll see. Let me just take the final and see what my opinion about it is, and if it's like, yeah, if it's too difficult, I'll definitely try to give you guys more material to study. Okay. Let's-
If you do a Zoom over the weekend, I'd be. Love to attend that.
yeah, for sure. Yeah. I think. Okay.
Okay, I'm looking over the slides,
Yeah.
and for me personally, I think the only one that's like still a little bit confusing to me is the solvent-based clearing versus aqueous-based clearing. In
Hmm.
like slide 3 and 4. Because I'm pretty sure that generally what clearing means is like making a tissue transparent for imaging. But I don't know, I don't get I was confused on the differences during her lecture.
Yeah, this is pretty. Yeah, I don't know too much about this kind of stuff too, but I know that one of the takeaways about clearing is that, we clear with molecules, and all molecules vibrate, and it's going to give off some kind of footprint on the Raman spectrum. And yeah, and so one of the main takeaways is that we want to make sure that the molecules that we're using in the clearing steps won't actually convolute the footprints that we want to look at.
Okay.
So if there's an overlapping peak over the CH region or whatever, then we should not use that. That's definitely one of the main takeaways about the clearing table. And I remember she actually also talked about how we can use, so it turns out that yeah, when we do these clearings, yeah, there are definitely reagents that create peaks that overlap the peaks that we're interested in. But then when we do stuff like the heavy water experiments, it's totally fine if we use those reagents that will have overlapping peaks with the other molecules, because at that point the deuterium is somewhere out in the cell silence region,
Yeah.
and so it won't interfere with that, so it's fine in those cases.
Uh,
I think those are the main takeaways. I don't know if she wants you to. I do remember that she specifically pointed out scale. I don't know if she wants you to go into that much depth. Yeah, I mean, if you are motivated, yeah, maybe you can actually look into these molecules a little bit. And but yeah, I mean, like you can tell that some of these molecules definitely have carbons and hydrogens on them, so
yeah.
they would interfere, yeah, with identifying molecules that weren't deteriorated or whatever. Yeah. So that's that. Yeah. So what are good candidates for clearing when you do Raman scattering? Yeah.
She has questions within the final.
Yeah.
Which I think is
Questions within the final exam?
Like,
the
yeah.
Oh yeah, yeah, yeah. Yeah. Yeah, I mean, I think she said that some of the questions, a lot of the questions on the midterm final are just going to be some derivative of these questions. And so, yeah, I mean, so when I take the final, I'm gonna, I'm just gonna be like, are the questions really similar to the questions on the review slides? And if they are, then I'm not gonna do anything. You guys should be, yeah, exactly. It should be fine. Yeah. Okay, so yeah. Bioorthogonal, that's click chemistry, that's like putting some kind of small tag. Yeah. Three small molecules is the alkyne. It's the, oh, I guess, I mean, you have them right here. And I guess the differences between these five is how they're incorporated. So this five I'm not even gonna try to pronounce it, but the one of them gets incorporated into DNA, one of them gets incorporated into RNA, amino acids, phospholipids, and triglycerides. So we have these specific molecules, yeah, that can associate with any biomolecule. Wait, did you guys say something? Something about? No. Yeah. Yeah, and this is just some examples. This is the Raman tag. I think this is for deuterium. I'm not sure, the Raman tag, or it might be one of these guys. I don't know. I think it's the deuterium, but I don't know. This is just the difference between the newly synthesized, that's shown in green, I believe. The D is for the deuterium, and then the yellow is the existing. Yeah, again, the newly synthesized proteins. This is kind of just a breakdown of like, oh, if we introduce deteriorated glucose, how does the deuterium, how is it distributed amongst the metabolites in the cell? Yeah, yeah. It was just based off of our analysis of metabolic pathways. Yeah, we can actually, it's cool. I think it's actually freaking super cool that we can be like, oh yeah, the hydrogen on the five carbon hydrogen is going to be replaced by deuterium. Yeah, I think that's pretty cool. I don't know. Yeah, yeah. And it's cool that we don't have to do the calculations for that. We can just like, yeah, use the prior knowledge. Maybe there's probably like software out there that allows you to do that.
Spectroscopy and seeing like how likely the probability is, and just now.
Yeah, yeah, yeah.
And then the software.
So yeah, I think again, this is just like, yeah, how the deuteriums are distributed, and then again, yeah, yeah. See, it's the C to D bond vibration is going to be captured in the cell silent region. And then fish. What does fish stand for?
Actually forget.
You know?
Slide?
Yeah,
No.
I mean, yeah, I mean,
From the slide. Fluorescence in situ hybridization. I was thinking the first one was fluorescence.
yeah. Do you guys, so yeah, do you guys know what a fish is now?
Wait, which slide? Which is that from the lecture 7? Or-
It's in the final.
It's in the
review.
final review, yeah.
Based off of the video, so I wanted to find that video.
And F is fluorescence. That's like some kind of fluorescence molecule.
Oh, that's from the guest lecture, I'm pretty sure.
Yeah.
Or was it-
She was presenting.
Oh, yeah, no. Okay, yeah. It was. Yeah, it's from the lecture 14. Yeah.
Took it out. Look at a tissue.
Yeah. In situ is, yeah, what does in situ mean?
I think it means like actually in like the organism.
Yeah, I think so too.
Not in the body, but it's like.
In the original place. Yeah. So I guess like
Where it is, so
Yeah.
you don't remove anything, you don't take it out of its natural environment, you can see how it interacts with. Well, maybe not natural, I shouldn't say that, but.
I think it's yeah, I mean like when we take the biopsy and then fix it to a slide, that's still considered in situ. Because we're freezing that sample in time. Yeah, that was once where it was once in its natural state. Yeah.
So you use this to label specific DNA sequences.
Yeah.
Yeah, you can do any, I mean, any nucleotide, right? You can do DNA, you can do RNA. Yeah. And a hybridization. Why is it hybridization?
I'm pretty sure it's because it's like when you use a probe and then it binds to wherever in the body, then like you can track the probe
Mhm.
and then see like I'm pretty sure hybridization is like when there's something already there and then you use like something you can physically mark so that it binds and then you see like where it binds to via the marker.
Right.
Because I'm pretty sure like the protein assays that we do like use hybridization, like the ones in like the midterm with like the red and yellow and green. Like I'm pretty sure that's via hybridization.
Okay, so I think hybridization comes from specifically the step in PCR, the hybridization step, right? Where I think it's the primers hybridize to the template. Yeah, so hybridization is that the probes that are used for fish, what they look like is they're a oligonucleotide, anywhere from like I want to say 17 to 100 base pairs long, and they're conjugated to some kind of fluorescent molecule. Those are our probes. And so when we fix our cells onto a slide, and then we introduce our fish probes, the probes are complementary to either a DNA specific DNA sequence or specific RNA sequence. It's a complement, so hybridize to that complement, and then we can excite our fluorescent marker, and that gives us the ability to see the location of the DNA or the RNA in subcellular resolution of the location of DNA and RNA. Yeah, that's what fish is. Yeah. Yeah, so in this image, I believe the glowing red things, those are the probes. And the green lines or the gray lines here, that's the RNA or DNA that's existing inside the cell. Yeah, you can use it to identify whether or not there is a specific mutation, or you can use it to either. Well, so it can be used in a binary fashion, where it's like, does my does my cell have this specific mutation or not? And then it can also be used to give you the subcellular localization information of typically RNA.
So the left is basically DNA fish, and the right is like using it as mRNA fish.
I mean, this on the right is also a little ambiguous because it's zooming into mitochondria, and mitochondria have their own DNA as well.
Okay, true.
Yeah. This must be an old slide because I believe that they use fish initially for just DNA, but then when they started to apply it to RNA, and they realized that, oh, actually we can get subcellular localization information by applying this technology to RNA. That's when fish blew up. Yeah, and now you have all these different derivatives of fish, like merfish and like all these. There's like crazy amounts of fish, and they're trying to, they're trying to. Whenever something gets hot, they always try to decrease. They try to decrease the throughput, make it as small as possible, and increase the number. So like I think with merfish, they're trying to be able to track over 100,000 different RNA molecules in a single snapshot using like the way that they combine different fluorescent markers together to give us different colors. And or one of the things that they do is like it's primer locking. So what they do is they have the fish probes actually, instead of just binding to one location of an RNA, it'll bind to two different locations of the RNA. And so then by getting a difference of colors, but then the thing is these two probes are designed so that they will actually bind to other RNA species as well. And so it's the combination of colors that will lead to the identification of that specific RNA species, as opposed to just being limited to a single color identifying, or a single probe identifying a single RNA sequence. Yeah. But yeah, yeah, so this question here, I asked her in class because I wanted her to clarify it to you guys, but then do you guys know what the answer is and why? So antisense, yeah.
Well, I feel like all of them.
It's wrong. It's wrong. And she said, I don't know if you guys were in class, but she said if you had picked all of them, then you would get 0 points. Well, B is definitely right. B is definitely right. But she said, I don't.
Oh wait, it's not C.
No, C is right.
Oh.
Yeah. So
wait,
I don't know if you guys were in class, but she said in these multiple choice questions, you can select multiple things. You can select multiple things. And I told her to be very transparent about the instructions for this. Yeah.
As long as it says like choose all that apply.
All that apply.
Just that kind of wording.
Yeah. Or else people won't know.
This, if you don't, then I assume you have to do two, because if there's two that are correct, and you can't just pick those two.
Right, yeah.
That's clear.
Yeah.
So B and C are correct.
B and C are correct, yeah.
It makes more sense because like anti-sense. It's like I can see that.
Maybe block the export, since like they're like synthesized RNA, but not like they probably have something on there where they can't get through the membrane.
Exactly.
Okay.
Exactly. That's the one that, so so D is what? It is one of the answers, or it's not one?
So it's like A, B, and C are right.
Yeah, and D's not.
Yeah.
And it's because of that. Can you,
Okay.
yeah, which is what
It's
again?
like the oligonucleotides are like synthesized.
Yeah.
So however they synthesize them, like they could probably attach something where they won't exit out of the membrane.
I think actually it's the opposite.
Oh.
So it's like, I think, okay, and maybe this is just kind of like where we are at with biology, but typically RNA, we don't see RNA penetrating the nucleus. It's unidirectional. So we get the export of RNA. RNA goes out of the nucleus.
Yeah.
Typically speaking, maybe someone's gonna come up with they're gonna come up with an experiment that says actually no, RNA is totally going inside. But And so that's why D is not the right answer. It's because, yeah, we can't, the penetration of RNA into the nucleus is like, it's not common, I guess. I don't know. It's not seen. Very common.
Wait, I thought,
Or is it? I don't know.
Wait, no,
Now I feel like I don't.
wait, mRNA is created in the nucleus and then exported out to like the cytoplasm.
Yeah, so it could, do its thing.
Mhm,
Yeah,
mhm.
but then it can't go back into.
It can't go back in.
Oh, okay.
That's what I'm saying. Yeah.
So blocking the export,
Yeah, because if you're blocking the export from the nucleus, wouldn't it be a mechanism that make it not able to go out?
Right.
which-
Right.
So like not the opposite.
Right. But then for that to occur, the the ASO has to be delivered inside of the nucleus.
Oh, I get it.
Yeah.
Okay, yes. So it's something the ASO needs to be able to go into the nucleus, but not leave. However
Yeah.
that works.
Yeah.
that wouldn't really happen because mRNA leaves.
Because
Oh,
mRNA.
I get
Yeah,
you.
exactly.
Wait, I get you now. Okay,
Okay.
because it should not be basically block the export. It means that it should not be able to leave.
Yeah.
But in order for it to get in inside, like you need to get it in.
Yeah.
So that would work.
Exactly.
Okay.
Yeah.
great.
Yeah.
I get it.
Okay. So also
I-
confirm that I have it. You can't get it in because the RNA doesn't go in.
The RNA doesn't go
Yeah.
into the nucleus. Let me actually. I'm gonna chat GPT
Oh,
this.
yeah, that makes sense now. Because I was confused, but that makes sense.
And RNA. Am I spelling nucleus right? I hope I am. Oh yeah. Okay. Yeah. Oh god. Okay. So maybe I'm wrong.
Not a structure.
But it's also micro and, wait, there.
No, but I think the reason.
Larger, it says larger RNA molecules such as mRNA stay in the cytoplasm.
Yeah.
Yeah, no, that's why I think the reason why D makes sense is because if you want a mechanism
Not
for it to lead, like to block, then you need to get it initially inside the nucleus.
Yeah.
But if it's blocking it, then it's not gonna be able to get it in the first place.
Mhm.
So I think.
Okay. I don't know.
Oh, but so
Yeah, says they can. I mean, yeah. I mean, the thing is, you said like something about modifying the RNA so that it doesn't go into the nucleus, and then, but actually, the reason why I said it's the opposite is because I said actually you probably need to modify the RNA so that it can get into the nucleus.
Yeah.
Because typically we don't see RNA going into the nucleus.
Okay.
So maybe
There's-
it's in that situation where you can get the mRNA to go into the nucleus, but it's not something that will occur naturally.
Right. Exactly.
Oh.
Or I mean, even that is up. I don't
If
know.
you get it,
I
if
don't
you
know.
get it to go in, then it can't block coming out, basically.
I
But
think
it
there
would
was.
stop it coming back out.
Right. I think so. I think okay, so this is my understanding. All of these mechanisms require all of these require proteins. So right. So for number one, to cause mRNA degradation, what happens is when you have a double-stranded RNA inside the cell, inside the cytoplasm, it actually the risk complex, AGO2, this protein, right? It has a biochemical affinity for double-stranded RNA species. And that actually what it does is it binds and it cuts it. So by introducing an ASO, what it does is it kind of attracts this double-stranded RNA cutting protein complex to cut it. And so that's what happens. When we talk about RNA splicing, altering RNA splicing, what happens is so maybe RNA can go into nucleus. I don't know what I'm talking about. D maybe, I don't know. Okay, so forget everything I said about D. Anyways, this is what happens. You have your pre-mRNA, right? And then you have your ASO come to like there's a junction between an exon and an intron. And so the way that splicing works is the spliceosome, another protein complex, it finds the junction between the exon and intron and intron and exon. It brings it together, it creates a lasso, and then it splices out the intron, right? But when we have an ASO that binds to one of the junctions, the spliceosome doesn't recognize that junction anymore, and so it doesn't splice anymore. What it does is what it might more preferably do is is span
Yeah.
that exon, span the next intron,
Yeah.
and then splice out that center exon entirely. And so that's how ASOs can alter RNA splicing. Yeah. So it's like preventing the binding of tRNA to mRNA during translation. This one, I don't know. I think I need to do a deeper dive on this, but it's like if you have the complement to an mRNA and it's being fed through ribosome, you know, the tRNA has a biochemical affinity to the codon that exists on the mRNA.
Yeah.
But if that's being blocked by ASO, then the tRNA is never gonna have that affinity, and it's not gonna happen. It's gonna stall translation. But the
Yeah.
problem I have with that one is like, how do we know that the antisense oligonucleotide is not being knocked off by the ribosome as it's being fed through?
Yeah.
I don't know. That one seems a little ambiguous to me. I feel like we didn't go over that mechanism to. So I don't know. And yeah, and D, I guess, yeah. I don't know. I guess what they're saying is, have antisense nucleotides been used in this manner to block export of mRNA from the nucleus? And maybe it hasn't. Maybe that's why it's not the answer. All right, maybe it's, I don't know. Maybe I'm gonna clarify with the professor, because I remember she said that, yeah, it wasn't the answer for this question. It was not E. But anyways, yeah. But yeah, let's look into that. Yeah. All right, let's move on to the next one. Okay, yeah. But yeah, how do you feel? You want to leave too?
Yeah, I think we can. I
Alright.
think that I actually need to watch that lecture because I haven't watched those days.
It's a good.
So I'll listen. Yeah, so I'll listen to her go over all the.
If I would, yeah, recommend studying, yeah, the past lecture and then probably Kunzang. And then also this, she had like 100 slides on spectroscopy.
Yeah, so know that.
I'm studying, yeah. And then you
Okay.
should be in good shape. I'll go over the final, and if it's like, holy shit, why are you doing this to students,
Yeah.
professor, then I'll put up some practice problems.
Okay.
Yeah.
And then, are you gonna talk to the TAs to do the clarification of the homework?
Yeah, I'll definitely do that. I'll do that like right now.
Thank
Cool.
you.
Yeah. Have a good weekend. Good luck for studying for finals, and yeah,
You have finals too?
I have finals. Yeah. Are you guys gonna take this is 168, right?
Yeah.
Are you guys gonna take 160 next quarter?
Yeah.
You are?
Oh, 168.
Yeah.
I'll be the TA for that class.
Do you know which section you are?
Yeah,
I'm gonna be a part of 2 different sections because my schedule's weird.
'Cause
11, 12, 20, is that one of them?
I'm in the 3 to 5.
5. I don't know yet. I don't know. I haven't picked my sections, but I know.
Those two.
Yeah.
Okay. Yeah. So what's gonna happen is like, since the section is, it's like Monday and Tuesdays, right? The
Yeah.
classes. They all conflict with one of my classes. So what I'm gonna do is like, I'll either take 2 of the Mondays or 2 of the Tuesdays.
Oh, I see.
Yeah, that's how I'll do it.
Oh, okay.
66% chance of,
Okay.
yeah, taking one of your classes. Yeah.
Hopefully.
I can teach you all. I can actually show you guys all the little tips and tricks that I've acquired from my experience.
Yeah, I'm excited for that class.
I only have like lab experience in my classes, which is like so different than, bio.
Yeah.
Do you guys? You're in Molly's lab, right? Wait, are you?
Marinium's lab.
Oh, you're in Serenity's lab. Okay.
It's bioinformatics.
Yeah.
So that's my like
Oh, so it's like.
experience. But,
You know?
Oh, so it's like.
Do you know Dasha? Or Daksha?
I do, but I don't really I've only met her a couple times. I didn't interact with her.
She's my classmate.
Oh.
She looks like freaking 11 from Stranger Things, right? She looks like 11 from Stranger Things. It's crazy.
Like, wait, I don't watch it, but I do know who 11 is. But
Yeah.
I only met her once. I don't
Okay.
remember her face.
Okay.
Thank you.
Okay. See you guys.
Bye.
Yeah. Good luck.